This is Unit 3, Cell Structure. So we first have to start about who are the scientists that actually discovered cells or parts of cells. So the very first one that we need to look at is Robert Hooke. Now, how you can remember him is Hooke took a look. Okay, what he actually looked at is he looked at cork. Cork is actually a tree, and he came up with the word. He actually coined the phrase cell. Okay, in Latin, cellia means a small room. Now, our next scientist was Leeuwenhoek. Leeuwenhoek actually used a actual, very simple microscope in order Now again, so Robert Hooke, he took a look. He was the first person to ever see cells, and he actually coined the phrase cells. Their cells. Now, these scientists actually helped Now, what was their contribution? What did Hooke do? Hooke took a look. Okay, Hooke was the first one to ever look at cells. Now, in order to So true or false? Robert Hooke was the first person to see cells. True. Bacteria cells have a cell membrane. True. So let's talk about how the first cell organelles as well. So they're the first ones to ever inhabit Earth. They're very simple. An example would be bacteria. Okay, so again, they do not have a nucleus, and their DNA is actually in a circular form compared to the double helix that immune system. Now, eukaryotic cell, they're much larger, they're much complex. They do have a nucleus, and they do have membrane-bound organelles, okay, in comparison to size, okay, so okay, that's a nice question that you need to think about. Now, our eukaryotic cells, they are basically divided into two sections. So you've got the main section, which is your nucleus. Okay, this is kind of a separate compartment, and this is where your DNA is going to be housed. Now, the cytoplasm is very fluid-like, okay, and it's the fluid portion, and it's outside of the nucleus, and this is where all of your organelles are going to be held. Okay, so it's separate from the external. Okay, so our actual plasma membrane layer. So how did prokaryotics and how did eukaryotics came to be? Okay, so we theorize that the mitochondria and the chloroplast have their own DNA, which we do know that is actually true. And it's completely separated from the nucleotide. Now, this actually supports the endosymbiotic theory, which basically states that eukaryotic cells actually evolved from prokaryotic cells by basically being engulfed or absorbed those other cells. So if we look, here's our ancient prokaryotic cells. So we have our nucleus, we have the ER, we have some of these different cells, and here comes our heterotrophic prokaryotic, or our aerobic, which means that they need air. Heterotrophic means they need to consume things. But again, it does not have a nucleus, so therefore, okay, it is still considered a prokaryotic. Well, here comes our engulfing that photosynthetic prokaryotic, okay, which basically means that this is the very. So let's talk about size. Okay, so there is a limiting factor, something that will deter or change our size, okay, and that's surface area. So basically, if the cell does not maintain a certain surface area to volume ratio, it can't transport nutrients and or waste fast enough through its cell membrane, and therefore it will die. Okay, so if we look, even though the surface area, okay, so the surface area to volume ratio actually decreases in size. Okay, so when we go up from our 1 millimeter to our 2 millimeter to our 4 millimeter, okay, the 4 millimeter is too large. It will not be able to transport enough nutrients and or waste through that cell membrane at a fast enough rate for it to be efficient. So again, if it cannot to be able to function. So therefore, instead of having just one cell, we break it into smaller cells, in which therefore will have enough surface area for it to be transported. Okay, so the actual large organelles have specific border. Now, what makes us different? One, what makes eukaryotic cells versus prokaryotic So all of the DNA, all of the instructions are going to be located inside the nucleus. The only time that it leaves. Now, so pretty much it is the same way that your nucleus is going to be your instructor. Okay, it's going to be the boss. It's going to tell now cytoskeleton. Okay, cytoskeleton is that framework, that basically the actual your filaments look like threads. Now centrioles, the only time you're going to see centrioles, or the only cell you're going to see it, ATP that they're going to some different ways that we use or create proteins. Okay, so endoplasmic reticulum. Now there's two different types of endoplasmic reticulum. If they do have ribosomes, it's going to be called the rough ER. If they do not have ribosomes, it's going to be the smooth ER. So if it's rough, it has ribosomes, it's going to make rough ER has ribosomes, and it's also going to produce protein. Okay, so both of these, okay, will either package or ship, basically can move or transport the Golgi. Now the Golgi body, basically it looks like a bunch of pancakes on top of each other. Now there's also some assisters that they have, okay, or facilitators that they have. Cell membrane if it's damaged. Now lysosomes, you might hear them as called suicide bags, basically because they can actually release their enzymes into the cytosol or the cytoplasm, where the cell okay, so, or damaged and killed, I should say. So if the cell is now mitochondria, okay, you've to have oxygen. Now your heart cells, okay, your heart muscle cells, your cardiac muscle cells, they act in order to function. So take a minute, look at this, and I also want you to fill this in for your graphic organizer. So true or false? All cells have nucleus. False. Prokaryotics do not. All cells have plant cells. Now we talked about animal cells. Okay, different differences. Okay, so go ahead and fill out your graphic organizer with the following labels. So there's a couple differences in plants and animals. Okay, so plants, they have chloroplasts. Chloroplasts act. So let's talk about the microscope. So focusing. .400x. Now we do not use the 400x because specimens. Now the electron microscope, there's two of them. We do not have this because if you notice, it is very large. Okay, it will not be able to fit in one room. It is basically looking at, it's called the scanning electron microscope, and it does not sad. To think of thin. Work area clean. Now on your graphic organizer, I want you to label these parts. So you have your body tube, nose piece, objective lens, stage clip, diaphragm, light, eyepiece, arm, stage, coarse focus, and fine focus, and lastly the base. So alright, so just some regular rules. What? Okay, so you always want to start with the objective lens, which is the scanning one. Okay, that's the lowest one. So you're going to use the coarse knob, which is the big knob, in order to find your actual image. Okay, so if you click it on your higher powers, you're not going to be able to see your image. Okay, okay, so focusing. Once you got it on the scanning, you switch to low. Okay, again, you can use the coarse knob in order to okay, so recap. Scanning is for coarse, low is for coarse, high is So, in order to see on high power, you find focus. Now, we're talking about drawing, okay? Make sure, try to use pencil, because otherwise, if you screw up, you're not going to be able to erase it. Okay, make sure they're labeled, and make sure they're clear, okay? So making a wet mount. This is really important when we are going to, basically it's anything really. I mean, this is what our so we don't want that. The same steps. So cleanup. Basically, store your microscope with your scanning. In that cabinet. Now, here are some common problems. Okay, so make sure you click it. Now, I want you to take the time in your graphic organizer, again, here are the exact same ones, okay? So when focusing a specimen, you should all diaphragm. It's kind of like alright, so let's talk about the cell membrane. Okay, so we also might hear it as the plasma membrane. Now, what exactly is it made of? Philic. That means they love. Okay, so it's designed by it's got two molecules. Okay, so it's phobic. Now, our cell membranes, specifically for us, okay, or eukaryotic cells, are selectively permeable. Basically, what that means is it's kind of like this bouncer here. Okay, it's going to let things, either they decide, let things in, and can come in versus. So if we look at the receptor is kind of like your receiver of your TV. Okay, it's going to receive chemical signals. Now your carrier, we also might hear it as a channel. Okay, it's going to membrane. Now your enzymes are very important, okay? They're going to aid in chemical reaction. That's the number one most important protein that we're going to talk about. Now anchoring actually attaches itself, okay, to that are going to harm us. Now, carbohydrates, okay, so again, carbohydrates are there. Most of them are attached to a protein, which is referred to as a glycoprotein. And pretty much what this does is the specific let's talk about passive transport. Now, passive transport means that it's any type of movement of molecules. It's going to go from a high concentration to a low. It also means that it does not need energy. Okay, so it's going to go through a concentration gradient. Okay, so if we look here, we have a large area or a lump of sugar, right? That's a osmosis is actually moving of water, okay? So now if we look here, okay, water follows so nothing happens. This is called lyse. Now our last one is hyper. Hyper means that the water is leaving the cell and therefore it's going to shrivel and shrink. Okay, so salt water. Okay, that is why it's so dangerous to consume salt water even though you're dehydrated. Okay, on a barrier island or whatnot. Okay, so again, let's look at plants. Now plants are a little different because they have that cell wall to protect them. So they don't actually explode or lyse when it comes into a hypotonic, okay, because what it actually does is it becomes very flaccid, which basically, or excuse me, it does not become flaccid, it becomes turgid. Okay, turgid actually means that it's really, really firm, okay, or stiff. Now, isotonic, that basically means that they'll start to limp and wilt a little bit, okay? But when it actually goes through a hyper, that's where it's now most of your so again, no energy, osmosis and diffusion, facilitated means it now why is the water moving to now opposite. This is like our pumps. Now let's talk about transport. Okay, so we have these vesicles, right? The vesicles that are associated with the Golgi, and they're going to move. Okay, it's kind of like the box for the UPS guy, okay, or gal, and they're actually going to move things either in or out. It's called exo. Now there's two different types of, or there's a form of endo, okay, which is called cell drinking. We call it Pinot.